In this experiment, students will learn about paper chromatography and ion exchange chromatography. Your kit should include chromatography chamber, packing tube, chromatography filter paper, dye mixture A for paper chromatography, dye mixture B for ion exchange chromatography, ion exchange beads, calibrated transfer pipettes, a graduated cylinder, and test tubes. You will also need scissors, water-soluble dyes such as markers, clear 70% isopropyl alcohol, rubbing alcohol, vinegar, glass cleaning solution, and distilled or deionized water. Activity 1. Preparing chromatography paper strips and chamber. 1. Use a clean pair of paper cutting scissors to carefully cut the sheet of filter paper, approximately 12 by 20 centimeters, into 6 individual strips. Two. Cut the bottom end of the chromatography paper strip to form a tapered end. 3. Draw a faint pencil line on each filter paper strip 1 centimeter above the tapered end. Step 4 is performed by the teacher. 4. Using scissors, the teacher should carefully cut 1 or 2 slits in the center of the top cap of the packing tube. The slit should be no more than 2.5 centimeters wide. Activity 2. Preparing acidic solvent A and basic solvent B. Acidic solvent A. Use the plastic gradient cylinder to prepare the first solvent as follows. Measure 50 milliliters of distilled or deionized water and transfer to a clean vessel. Measure 50 milliliters of clear rubbing alcohol, isopropyl alcohol, and add it to the water. Using a calibrated transfer pipette, measure 10 milliliters of vinegar and add it to the water and alcohol. Gently stir the mixture. Label this solvent acidic solvent A. Wash the graduated cylinder with distilled or deionized water. Basic solvent B. Use the graduated cylinder to prepare the second solvent as follows. Measure 50 milliliters of distilled or deionized water and transfer to a clean vessel. Measure 50 milliliters of clear rubbing alcohol, isopropyl alcohol, and add it to the water. Using a calibrated transfer pipette, measure 10 milliliters of window cleaning solution, such as Windex, and add it to the water and alcohol. Gently stir the mixture. Label this solvent basic solvent B. Wash the graduated cylinder with distilled or deionized water. Activity 3. Dye separation by paper chromatography using an acidic solvent. 1. Place the tip of a transfer pipette in dye mixture A. Let the sample solution be absorbed by capillary action into approximately half of the thin tip of the transfer pipette. Squeeze the bulb very gently. 2. Place the tip of the transfer pipette onto the filter paper at the pencil line. Very gently squeeze the bulb and allow the sample to be absorbed into the filter paper until it forms a small spot. 3. Let the sample spot air dry. 4. Repeat steps 1 through 3 for a second strip. 5. Transfer all of the acidic solvent A to the chromatography chamber. 6. Attach the top cap to the chamber and lower the paper strip to immerse in the solvent at the bottom of the chamber. Only half of the tapered tip should be immersed in the solvent. 7. Once the paper strip is in place, fold the top of the paper strip to keep it from slipping. Tape it on the top of the cap. 8. After approximately 10 to 20 minutes, observe what happens to the sample spot. What colors are separated from the sample? What is the order of the colors from top to bottom? 9. Remove the paper and let it air dry. Note the colors. 10. When finished, discard the solvent with water down the sink and rinse the chamber with water. Activity 6. Separation of a mixture of dyes by ion exchange chromatography. Experiment procedure. The solution made up of 2 dyes, sample B, is added to the ion exchange beads. One of the 2 colors will bind to the ion exchange beads that will settle to the bottom, and the second dye will remain in the solution. This will change the color of the solution. 1. With a calibrated transfer pipette, remove excess water from the suspended bead solution. Just leave a small layer above the beads. 2. Add 4 milliliters of the diluted dye mixture solution to the conical tubes. Note the purple color of the dye sample. Cap the tube with caps provided. 3. The dye mixture is made up of red and blue dyes. Shake the tube vigorously over several minutes. Note the color change. 4. Let the beads settle. 5. After 5 to 10 minutes, shake the tube again. 6. Note the color change. Conclusion. You have just learned how to separate molecules using paper chromatography and ion exchange chromatography. Both procedures are used in research and industry laboratories. Paper chromatography is used to separate small molecules, such as dyes, and ion exchange chromatography can be used to separate biologic molecules, such as proteins, from DNA. We hope you've enjoyed DNA Depot Experiment 401, Separating Color Pigments by Chromatography.